Hello, my name is Mike Ward. I'm the Global Director of Content at Pink Sheet, Scrip and In Vivo. We're here at the Biotech Showcase 2018, a meeting that runs in parallel with the JP Morgan Healthcare Conference, a conference which typically kicks off the year for the pharmaceutical industry, where pharmaceutical companies, biotech companies, and investors all get to meet each other and to sort of discuss the issues that are facing them, but also to look for opportunities where they can work together. One of the sort of the hot areas is in oncology, and also we've seen in recent years the re-emergence or the renaissance of gene therapy. I'm joined by Dror Harats, who's the CEO of an Israeli biotech company, VBL Therapeutics. So Dror, you're actually in that gene therapy space. So could you describe what it is that VBL does?
Yes, of course. So we are in the gene therapy space, and actually we are giving a drug which is a very sophisticated drug. We are actually giving an information. We are injecting this DNA piece through an IV infusion. So it's a simple IV drug like every other IV drug that patients get. And this getting into tumors, no matter wherever these tumors are. And actually it's doing two ways. It's working in two ways. It's destroying the vasculature of the tumor and actually starving the tumor, but it's also induced the immune system to recognize the tumor within the tumor itself. So what we do see in many different solid organ tumors like brain tumor, ovarian tumor, lung tumor, thyroid tumor, so that we can have an effect on the tumor itself, but the major thing that we prolong the life of the patients, so we have a major effect on their survival.
So how do you disrupt the vasculature? Is it like VEGF where it actually stops, blood vessel proliferation?
That's an excellent question, and thank you for asking it. So the way we work is a completely different way than the other drugs like anti-VEGF. The tumor has many ways to build new blood vessels, actually more than 20 ways. So if you just destroy the VEGF, the tumor will find a way to work around it. The way that we work, we put a deadly gene within the vasculature of the tumor. And the moment that the tumor tries to build the new blood vessels, this deadly gene, like a poison, is getting activated. When it's getting activated, it's destroyed the blood vessels of the tumor. So we don't care how the tumor is building its own blood vessels. It can be VEGF, it could be something else. No matter what, we are not going to enable the tumor to keep on growing.
And is it sort of specific to particular tumors, the way it works, or can it work on any tumor?
So actually it can work on almost every tumor. Every tumor that build up like an organ, like a solid tumor, need blood supply. All of them have what we call angiogenesis, so our drug will work there. And when it works there, it destroys the blood vessels, it induces what we call neoantigens, but because we have a virus there, and the immune system is geared to fight viruses, it's actually fighting the virus and the tumor cells. And that's how we actually kill the tumor for a long period of time, because we induce the immune system now to recognize the tumor. And then it can work for a very long time and prolonged life.
Okay, and where was the technology originally developed?
So the technology actually developed within the company itself in Israel. We're a public company here listed on the NASDAQ, but we are an Israeli company. It's all started years ago in my visiting professorship studies, and then we developed it. And the way that we are doing it is actually we are using what nature is using. So we are using a piece of DNA, like all the genomic that we have in the body. We are using a very sophisticated piece that we modified in a way that now it's very specific to the tumor vascular. But we also use a virus because that's the best way to actually put genes into cells. So the whole drug is actually a virus with information in it.
And what's the virus? Is it a adenovirus?
It's an adenovirus. It's a simple flu-like virus. It actually does not replicate, so it cannot give you a disease. It just gives the information. And it does not integrate into the genome, so there are no risk of creating any disease.
Okay. So the technology. So you say you have drugs. What is the sort of status of the programs you have?
Okay. So we are now at the end of a phase 3 pivotal trial that was done under an SPA with the FDA. So it's actually a registration single trial that is going to be end very soon, and we are expecting top-line data in the first quarter of 2018, so it's just around.
And which tumor are you going for in that case?
In the brain tumor, we're going after glioblastoma, which is the most devastating brain tumor, and that's after having a successful phase 2 trial where we showed dose response in GBM. We also had a successful phase 2 trials in thyroid cancer and in ovarian cancer, and that's why we started another pivotal trial in ovarian cancer just this month.
Okay, so you've got 2 programs. Are you doing this on your own, or have you got partnerships?
So actually we are doing it on our own, and we managed to finance it quite well. The deal that we did was just a geographic deal in Japan for VB111. All the rest we are doing ourselves right now.
So, and who's that with? With the Japanese?
Nanocarrier, which is a group that's actually working on gene therapy and gene technology as well.
Okay. So you've got 2 programs. Any other programs?
Oh, actually we built a real small biopharma company because we have a great pipeline beside of the VB111, which is our lead compound going into this different indication. We have 2 other platform technologies. One is a small molecule for inflammation, and the other one, which is very interesting, is actually a group of antibodies, different antibodies that working on a very unique protein that we discover, which is a surface protein, which has actually a very important role in the ability of cells to wander in the body. So it's very crucial for inflammation, but also for the metastatic development of tumors.
So your company's actually got the full arsenal of a gene therapy, a small molecule approach, and also antibodies.
So I was saying that this is a real small biopharma companies going after unique targets.
Right, okay, so you haven't got any partners, or you have the nanocarrier partner, but how are you sort of financing these programs then?
So we were financing it first through VCs and investors as a private company. Later on we had our IPO and 2 follow-ons, and we are well financed. We have, at the time of the data, we expect to have more than $50 million in the bank, which actually give us more than 2 years ahead of time. But we are going to be here opportunistic because this is going to be such a big drug for many different indications in oncology. So we can develop it by ourselves. We can go along and develop it and sell it, but we also will be open to partnership at this point.
So, and how are you building out your own commercial infrastructure then?
So actually we hired Dr. Corina Epperly, that is ex-BMS, and she was actually working on drug development here in the United States. One of them is Optivo, and that was actually aimed for GBM as well, so she's quite familiar with the field, and we are building our own force on ground here.
So, right, what, in the US?
In the US.
So you're building out that. So at this meeting, where, as I say, we have pharmaceutical companies, we have other biotech companies, and we also have investors. Who is it you're looking out to reach out to?
So actually the potential strategic partners, which is a big pharma or big biotech, were I wouldn't say our target, we were their target in this case, because they know that we are expecting top-line data. So we had quite good meetings. Of course, we weren't ready for any deal at this point. We're waiting for the data. We had very good meetings with investors, as you can imagine, with analysts and with bankers, with the whole industry.
Right, okay. Great. Well, Dror, thanks very much for coming by and telling us the story.
Thank you very much.